Okay, so here's the big picture, and it is big. It's basically, this is a project I started 3 years ago, and the ultimate goal is to monitor the health of the biosphere, which means all life on planet Earth. That's rather ambitious, right? Um, and think of it as basically putting smoke detectors around the whole world, but instead of looking for smoke, we're monitoring the health of the biologics of the Earth. And to do this, we're looking at plankton. And you're biologists, so you, unlike most people, probably know what plankton is. But just to refresh your memory, plankton is basically a general term for anything that floats in water that can't swim against the stream. Usually you think of tiny creatures, but jellyfish are actually plankton because they can't swim against the stream. And so these creatures are amazing. They provide up to about 2/3 of the oxygen we breathe. They sequester the largest amount of carbon from the atmosphere, taking CO2 and burying it in their dead bodies at the bottom of the ocean. And also they're the baby food to practically every species of fish. So I tell people, if you like sushi or you like to breathe, you got to thank plankton. So the basic theory is, let's monitor the health of the plankton because they're basically like the classical canary in the coal mine, which is called a sentinel species, a species that you look at to see if they're in trouble, you're going to be in trouble soon. So that's the premise of all this work, is to monitor the health of the environment by monitoring plankton in situ, meaning where they live, not in the lab. And so that's the goal. And the challenge is there are about 4,000 species of plankton. And so it's really hard to train a machine learning algorithm on 4,000 species because you need a great training set. But nature has been kind. At any particular one location where we would have a sensor, there are only about up to 24 species. So when we get into classification, that'll be an important factoid. So these images were captured from a microscope I developed. And it's a very simple microscope. There's no lens, there's no optics, there's no focus, which makes it very easy. You basically put the plankton right on top of an image sensor, which is basically a camera without a lens. And then you shine a light to look at its shadows. And this is the apparatus on the left. Unfortunately, we're not going to be building at this class, but we're going to use data that was generated from this microscope. So you're basically going to see plankton swimming on top of an image sensor. And sometimes the image will look fuzzy, especially the further away it is from the image sensor. And so we've made actually a version 2, which uses a laser to make holograms. So that, if we might get to that, that adds more resolution but more complexity to the problem. So again, the problem is to monitor plankton in their environment and to classify them, to count how many they are and what types they are. And also something more subtle is to look at their movement. If I come into work one day and I'm limping, you know I'm still alive, but you know something happened to me. And I might say, oh, I was horseback riding or I was skiing. You know I've done something to injure myself. And you can tell that by just how I move. We want to do a similar thing with plankton. Because if they change their swimming behavior, that affects their ability to eat, to escape predators, and to reproduce. So those subtle, non-lethal injuries can have cascading effect on the health of the biosphere. So this is a video. It might be choppy because of our internet connections, but you have this video. It's only 10 seconds. And this is going to be the first video you're going to analyze. These, I hope you can see it. These are plankton I put right on top of the microscope. And right away, you can probably see there's at least 3 different species. Clearly the big sausage ones here, those are Blepharisma. Then these tiny little ones that are fluttering about, those are Acilia. And then these clumps, I call those the salad bar. These are algae, clumps of algae. And to get a perspective, you're actually underneath the microscope looking up. So things that are closer are right on the glass. The algae is actually floating on top of the drop of water. So they're the furthest away. And the further away they are, the fuzzier they get. So this course, you're going to be able to download this video, separate all the items. That's called detection. You're going to track their movement, and you're going to do classification on them. You will have the skills to do all that by the end of the term. And so I'm going to quickly breeze through the different functions you're going to learn how to do and what they and the purpose of them. So detection is the first thing you're going to learn how to do, which is to separate the object from the background. And in your career, I'm sure you're going to work with microscopic images from confocal microscopes, light field, dark field microscopes. The same algorithms you're going to learn in this class, you can apply to that. And you'll probably know more than you'll do in your career because people usually just use static images. You're going to be analyzing video.